I'd call, I'd email, I'd write, nada, not getting back to me at all. So why am I telling you this? Well, because I want you to focus on that first part. Okay, the first part is I want you to understand what this feels like to us. I know more now. I think we know more, so much more about our disease and about our gene and what it does. We know what to ask for. And that wave of emotion that I felt when I was on the phone with my wife, it's like this sense of relief. It's like this bursting of a dam inside of us. I wish you could see me in my little home office, like the scientists in the room. Like, I hope you understand what happens. Like, I've emailed you, and then there's an email in my inbox, and it chimes, and you're responding to me, and you're agreeing to be part of this, and you're throwing your hat in the ring with us, and you're joining our tribe. And so when I get that email, that really matters. Like, I laugh out loud when that happens. Sometimes I cry with joy. Like, my wife and I both celebrate that together. And the mere fact that now in those emails and in the conversations that follow, we're using words like activation. We're talking about disease models, biomarkers. Every time I still get emotional about that. I want to share something else with you before we get started for today. We have a Facebook group, and it's for USP7 families. Not everybody that's been discovered so far in the world is part of the Facebook group, but it's very active. We now have more than 265 patients that we know of around the world. That's in 30 US states and 28 countries. And people get into this Facebook group, and I can tell they've never been on Facebook before. That's just, they join specifically for this group. That's why they come in. And that's because they're desperate for information. They really want to know. They have a million questions. They want to know about medications. They want to talk about seizures. They want to know about behaviors at school. Does anybody else have this? What have you done for that? How about anxiety, GI stuff, surgeries, recovery after those surgeries, pain threshold, I mean, you name it. There's so many things in this group. And I've been doing this for 8 years, and I've never seen so much raw thirst in our group for information. So I have amazing news for you, the scientists in this room. These families are reading your papers. They are reading what you write, like at your institution, in your lab, at your desk. I don't know whether it's ever crossed your mind that moms and dads and patients themselves all over the world are going to read your words, and they're going to look them all up and weigh them and debate them and put so much hope in the sentences that you write. If you ever find it difficult to go to work, like you wake up in the morning and you're wondering, do I make a difference? Is my work making a difference? I just have to tell you, yes, 1,000,000% yes, you are making a huge difference. I want to thank you all for being here, for making the trip, for making yourself available, for being willing to talk to us, for doing this work on our gene and our disease. If you can hang out today, you don't have to rush out of here and catch a flight. Please make some time to talk to some of the families that are here, to talk to them, to answer their questions, to hear about what they want to know. We really appreciate what you do, and we're so grateful to you. Thank you.
Everyone, I'm so glad that you're all here today. Very excited to see the turnout. I'm Becky Rotz. If you didn't know, that's me. I've talked to so many people over the phone, so now it's nice to put all of your names and your faces together. I just have a couple things to kind of talk about today. Well, first, thank you so much to all of our sponsors that we have for our meeting, because we definitely couldn't do it without you. And sorry, I don't have my mouse, and the touchpad is like driving me crazy. So this is really, really, really good news. We now have an ICD-10 code. It's going to be active October 1st. So your physicians, when you go there and you are talking to them and they're using a diagnosis code, this is the new code, Q87.87. And it's extremely exciting that we've gotten that, and we went through meetings with the, CDC, and we sat in and we had to present material to them, and, it was very exciting that it was on this year's, list of new codes. So make sure when you go to your next doctor's appointment, you let them know, and they can start using this diagnosis code. Also, Matrix. We really would love to have every single family join Matrix. Matrix is a way that we can have surveys about our patient population, and it really gives us a lot of really good information. But not only just that, researchers can then look at this information. It's de-identified, so they won't know who the individual person is, but they can see how they're answering these questions on these surveys. The surveys might be about neurology or GI. There's a variety of surveys. Also, when we give blood to our biorepository, we need you in Matrix and have your information in there. And I put up here the QR code and the link for you to sign up. And if you need that later, just come and see me. So Dr. Schaaf is here, and we are so excited that he's here. He did a big phenotyping study, and I believe that's going to be what you're talking about while you're here. So we are so happy that he can share the results of that and talk to all of you, talk to our patients, see our patients. He has absolutely, without a doubt, seen more Hao-Fountain Syndrome patients than any other doctor on the planet. So he is the expert, and we are so happy that he was able to come all the way from Heidelberg to join us. We have some needs. We need a clinical center of excellence, somewhere that can be the people in the US, the physicians in the US that kind of look at Hao-Fountain patients. Now, I would say Florida and Texas are the two states where we have the most number of patients, but really kind of Texas East is very heavily where most of our patients are located, that we know of where they are. But if we can get someplace in Florida or Texas where we have, 10 or 12 patients in each of those states, that would be kind of nice. So if you know of a physician, or if you know of a clinic that would be willing to see, even if it's not in those states, if you know of any place, if we can go to somewhere where we can start sending patients, where one physician or two physicians look at them and start seeing our kids, that would be very helpful. And then every single month we have a monthly virtual lab meeting for scientists. And these are so helpful, I think, for everybody in our community. And it's really great for the scientists. We've seen so much collaboration out of it. If you are a scientist and you would like to pick a date and put it on the calendar, you can come let me know what date you would like to host a monthly virtual lab to share your work, present your work, and we are more than welcome to put you on the calendar. Oh my goodness. I think that was everything. That's why it wasn't going any further. So next we are going to go ahead and move on to our next presenter. We do have a virtual program. So if you want to look at the virtual program, you can see that QR code, so you can look at who the next speakers are. And I'm going to just try to pull it up on here too. Okay, so next up is going to be Mandan.
Hey, good morning. It's so quiet. they make the West Coast guy talk first. It's like 6'8". Anybody here?
Yeah, wherever you're comfortable.
All right, I might move around a little bit. So my name is Mandon Foley, and I am a parent of 3. Over there, we've got Maylee, who's my 16-year-old, Hendrix, that right there with the blue hat on. Stand up, buddy. Both of you. There we go. That's Hendrix Foley. He's the one that the reason why we're here, and he's been in some of the papers you guys have researched, and we're glad that we've been able to be a part of that. What's going on over here? I can see people looking at it. I was asked to talk. This is a parent perspective, so wanted to give you just a little bit of perspective from our family. And so I got into life pretty early as far as adulting, and I was a young entrepreneur. I started a business. I'm in the insurance business, and have 3 kids. So father of 3. My oldest is 18. He's going off to college now, and married like 19 years, and in early 20s decided to have some children. And your neurotypical children, had a boy, had a girl, and we thought we had it figured out. We even had some stumbles along the way. Our 18-year-old now, he was born 7 weeks early and spent the first year of his life in the NICU. And my wife went into labor early, visiting her parents in Idaho. It was December, so we drove home 13 hours in the snow with our newborn baby attached to an oxygen tank in the back of the rented car. And it was quite nerve-wracking. It sounds like the story of our parents going to school, uphill in the snow and everything. But that was our first baby experience. We hadn't even gone to the Moz class, if they still have those. None of that. And so we just we figured it out. And we're a young, driven couple, entrepreneur, thought we had it all figured out. And then we had Maylee 2 years later. So we had the boy, the girl, and there are a lot of books and there are a lot of things to help you along the way if you don't know what the heck you're doing. And we didn't, but we just figured it out. And so, of course, 7 years later, when we decided to have another baby, we had the one that there's no book for. And so what I wanted to talk to you guys about was being comfortable in the unknown. So, and everything that you guys are doing here, it's helping us change that so that it is more known. And we talked about the Facebook group. There wasn't one. I think Hendrix was 54. Yeah, it's kind of interesting to have a number, and 100 more people, joined since that. When I was thinking of what am I going to talk about, and I've done a lot of self-help journeys. You look through my Audible, and that is a sad man. And no, I'm trying to figure stuff out, and I've had a lot of fun doing some hippie stuff, doing some diet changes and exercise and everything. And part of my journey, I had about a year ago, I was in this self-help meeting, and we wrote thank you notes to our children about what we're thankful for. And so I'm going to read this to you. I wrote this to Hendrix. They have no idea. This is, thank you, Hendrix, for teaching me how to unconditionally love someone. The beauty of it is you didn't even know you did anything. You exposed me to love with no expectations. It showed me how to love someone in the moment, in the present, the now, no matter where they are in life or how I think that they should be doing it. It's made a profound difference on my view of the world. And I thank you for the influence of my journey. I love you. love you, Dad. I love you, comma. Or comma there. I love you. And so the profound part of that for me was the love with no expectations. And as parents, we don't know that we have these expectations on our kids, but they could be as small as learning how to crawl, as, you know, walk. And you start missing some of those things. And you're like, oh, what's going on? And we thought we taught our two older kids how to walk, you know, how to crawl. Might have tried to bribe them with some bottle or binky a couple feet from them and tried to encourage them, show them where to put their feet and their legs. And all we really did was observe a neurotypical child figure it out themselves and thought that we did something, and we didn't. And so that was kind of a little bit of a mind-blowing realization for me. I thought I had more of an impact in there, but apparently kids have this innate, like, driven growth part of them that they're going to figure it out anyway. And so, so what I learned how to do is just celebrate every single little win without an expectation. And I didn't realize what I was doing with the older kids, because you expect them to learn how to walk just like all the other kids. And then you brag to other parents about, you know, oh yeah, he learned how to talk. But it was, you know, 18 months old. Whatever that may be, I missed so many opportunities with my oldest kids, you know, because of little expectations that I didn't even know that I had. And so in a way, I wish that it would have went backwards in order. I wouldn't change anything at all, but it opened my eyes to see things differently. See things differently when I'm hearing all of your stories. I see everything differently because my expectations are pretty well non-existent. And so it allows me to have the freedom to love every moment and celebrate every single win. So, and I'm not getting upset right now when he's running across the room, going somewhere. I have no idea. Because, you know, the new me, he's going to the bathroom. All right, we learned some sign language in this process too. But I think it's adorable, instead of thinking that I have any control of what's going on in this world. I'm just loving every bit of it. And so I would encourage you guys to do the same thing. I thank all of you guys for being here and being part of changing some of the unknown, so that we have something to help the new parents and the new patients that join the family. So.
So we're going to start our scientific presentations today. Dr. Ryder was going to be here, but he couldn't make it. But Dr. Hedgecock, Taylor Hedgecock, correct? Oh, it's a Mr., not Doctor yet? Not yet. Okay. So Taylor is going to go ahead and do the presentation.
Hi, I'm Taylor Hedgecock from the Tulane University School of Medicine. I am presenting today in place of my PI, Dr. Florence D. Ryder. I'll be talking about modulating circadian rhythm through USB7 targeting small molecules. So Hao-Fountain Syndrome is a rare neurogenetic disorder that is caused by mutations in the gene USB7. And it's characterized by autism spectrum disorder, hypotonia, which is low muscle tone, and epilepsy. And in 47% of cases, there are sleep disturbances. And this leads to a question of, is there a possible circadian rhythm defect? So circadian rhythm and human disease sort of go hand in hand. Circadian rhythm is a biological process in which cells maintain their homeostasis with these 24-hour oscillations of rises and falls. And in disease context, circadian rhythm is known to be affected. And in neurological disorders such as Parkinson's, Alzheimer's, sleep-wake disorders such as narcolepsy, and even with cardiovascular disorders, pacemaker activity is innately circadian rhythm linked. And in the normal state, you have these robust amplitudes of rises and falls over a 24-hour period. And in disease states, you can either have a reduction of these rises and falls, which is a lower robust signal, or you have a shortened period in which instead of 24 hours, let's say it's 20 hours. So the clock is speeding up in the cell. And our lab has been working on a circadian rhythm model, which I'll try my best to show here. We'll start here with CRY1 and PER2. These are known as the negative repressors of the circadian clock. They inhibit BMAL and clock complex, which is a core clock component which activate transcription factors and helps maintain sort of like a timing chain on an engine, helps everything kind of progress along like it should. And whenever BMAL and clock, they control the transcription of CRY1 and PER2. And as proteins keep getting synthesized, you have a accumulation of CRY1 and PER2 protein, which end up getting shuttled back to the nucleus to inhibit that complex. And the cycle kind of keeps circling back and forth like that. And you see this little guy called MAGEL2. This is a ubiquitinase, which targets CRY1. And it leads to its degradation via the ubiquitin proteasome system. And it is involved in kind of fine-tuning the circadian rhythm. And you can see USB7 shows up here. And it is the opposing deubiquitinase for CRY1, which leads to its stability. So MAGEL2 helps reduce CRY1, USB7 helps to stabilize it and keep it around. And so how our lab has come to this model, we work with what's called, sorry, I forgot about these little animations. So MAGEL2 is involved in another rare neurogenetic disorder, which is called Hao-Fountain Syndrome. They also exhibit sleep problems. And as we know, Hao-Fountain is caused by mutations in USB7. So our lab generated this circadian model by using what are called dental pulp stem cells. And these are neural crest stem cells that are derived from baby teeth. And they are multipotent, which means they can be turned into any type of tissue. And that includes neurons, glia, adipocytes, which are basically fat cells. And they are easy to collect. Babies and like young children lose their teeth as they age. And so it's easy to collect versus like, let's say, iPSCs, which sometimes require biopsies. And we can take samples from long distances and transport them with kits, which I'll talk about at the end of the talk today. And we're able to collect these from family members because a tooth falls out, it's simple as picking it up. And you can see in this table over here, the long list of disorders that our lab has accumulated over the years. And we have 2 individuals in our repository with Hao-Fountain Syndrome. So the stem cells are grown from what's called the dental pulp, which is the internal component of the baby teeth. And we get this from both patients and neurotypical controls. And our lab has experience in turning these into various different tissue types. Notably, since Hao-Fountain is a neurogenetic disorder, we can turn them into neuronal cultures. And to date, we have 2 Hao-Fountain syndrome lines in our lab. So how do we measure circadian rhythm in patient cells? Well, we take our DPSCs and we introduce a PER2 luciferase reporter via a lentiviral vector. And we select and stably transduce these lines to make these reporter lines for this core clock component. We then turn the DPSCs into neurons, plate them on a 96-well format, and we put them on an automated biospa system, which is essentially an incubator with a robot arm that picks up the plate, moves it over to the plate reader, and takes readings for every 30 minutes, and we can do it from 2 to 4 days. So that's a lot of data and readout from live cells we can acquire. And notably, we can then turn it into this readout of bioluminescence for circadian rhythm and discern differences. So our lab has experience in circadian rhythms with another rare neurogenetic disorder called Prader-Willi syndrome, and it experiences excessive daytime sleepiness. And we found in our lab that Prader-Willi deletion individuals, specifically neurons, showed a shortened circadian period phenotype compared to their neurotypical controls. And not only was there a defect, we were able to rescue this defect using longdazin, which is a CK1 inhibitor, and we were able to make that shortened phenotype long again. So we were able to find the defect using our models, and then subsequently show we can rescue a defect. I know this looks like a bunch of squiggly spaghetti here. We've been working on optimizing it, and I'll show you our progress in a couple slides from now. But our collaborator, Dr. Andrew Liu at the University of Florida, has currently tested 2 USB7 inhibiting compounds, GNE 6640 and XL177A. One's an allosteric inhibitor, and the other is a catalytic domain inhibitor. And he was able to show that both compounds were able to decrease that amplitude, essentially weakening the circadian rhythm's robustness. And in our lab, we've been optimizing this for over a year now, and we've finally gotten to the point where we have the beautiful curves that our collaborator Andrew is able to get. We're working with another inhibitor called P22077, which is another USB7 inhibitor through the catalytic domain. And we're working on optimizing the concentrations for both this as well as GNE and XL177A in our system to show that we're able to not only detect these changes, but also show we can significantly reduce the circadian rhythm through this Cry1 USB7 interplay here. And our future directions with this research is to take our 2 Hao-Fountain Syndrome lines and introduce that PER2 reporter and monitor, like, what kind of defects are there? Are there period defects like Prader-Willi syndrome? Are there amplitude defects with Cry1? What are there? And that's what we're going to look at. And we're going to also introduce Hao-Fountain syndrome mutations of USB7 to those US or U2S lines, which the previous slide showed, and see how do these mutations affect the period or amplitude or anything circadian related. And further on down, we'll treat both patients and transfected cell lines with these inhibitors and see if we're able to either rescue or perturb the system, just to see if there's any changes we can detect and potentially fix if we can. And I kind of talked through it pretty quick, but I'd like to thank my lab as well as my undergrad Margot, who's been instrumental in optimizing the system for detecting these circadian readouts and getting those beautiful rises and drops of curves for the Cry1. And I want to end today with being involved in our research with our DPSC kits. So enrollment in our research study just requires the completion of this Qualtrics survey at this QR link here. And upon completion, We will ship out a DPSC kit free of charge that'll have transport media, basically for the tooth to have nutrients as it's in transit. Parafilm, which is essentially saran wrap, which will wrap around the tube to prevent leaks during transit, and a biohazard bag and a return label, all free of charge to anyone who finishes the survey. And if you have any other questions about our research, feel free to reach out to my PI, Dr. Ryder, for any more questions. And that's my presentation today.
You mentioned baby teeth, but I assume you take wisdom teeth too, right?
Yes, our lab has a couple of wisdom teeth in our repository, but we mainly go for the baby teeth. Because the best part is since everybody loses the teeth about the same time, everybody's almost age matched. So we won't have to worry about age matching as much.
I'm just thinking for adults in the group.
Yeah.
That's a way they can then they're.
Yeah, we have got I believe, I don't want to put an exact number, but we have wisdom teeth in our repository. Not sure how many though. Yes.
I'm curious whether it would help you guys add more teeth from our group.
Yes, and I wanted to include that survey here is just reach out and like advertise because we need more samples. 2 is better than none, but more than 2 is greatly appreciated to help progress things.
Is your hair?
Okay.
I don't. I will wait too long.
Yes.
America pack. So I did yes. Who will told how I was idea I'm going to buy something. What?
All right, next up, I'd like to introduce Boris Bonaventure from Mount Sinai.
Can you guys hear me? Morning, everyone. Thank you so much for coming here. Thank you to the foundation to inviting me to present this data sets we have since reaching this year. So my name is Boris. I'm researcher, assistant professor in Mount Sinai Hospital in the team working on virus-host interaction and antiviral immunity. And I reached to the foundation 6 months ago because of our data we got on the function of USP7 in human immunity against viruses. And I'm gonna present every aspect of this project, showing that USP7 could have also antiviral function against viruses. And if you have any questions, any remarks or feedback to give us regarding like immunity of the child and the patients, feel free and let's discuss about that. So before starting, so you're gonna see that the main take-home message of this presentation is basically USP7 depletion could lead to higher infection rate. But before starting on the digging on the data, I should really disclaim that what we have now are preliminary data. We use like model cell lines that are used in the lab that are not exactly the same as primary cells, so it doesn't recapitulate exactly what happened in human. We use like hard depletion of USP7, it doesn't mimic what happened with the mutation with the kids. We are using viruses that we grow in the lab that we amplify, so they are lab adapted, they are not the same that circulate in nature, for example, so that makes a difference. And also something important when we look at viral production or viral infection is something different than looking at the disease and pathogenesis. It's not because you have a higher viral loads that you have a higher disease and higher pathogenesis. It's different, it's connected, but to really conclude about that, we need in vivo data that we don't have so far. So everything is preliminary and we don't have like firm confirmation of what can happen in animal model, for example. But if you have any interest to discuss about that later, please feel free to reach me. So this is the lab. At the left side, we have Dr. Jeffrey Johnson. He is specialized in the use of mass spectrometry approaches and omics approaches to study virus-host interaction. We are two researchers, myself and Marcos Martin, in the lab working on virus-host interaction, two PhD students, Trevor and Andrew, two technicians, Heng and Kina, that helps me a lot to get this work going together. So as I said, the lab is mainly focused on using mass spectrometry approaches to study virus-host interaction. And basically, our typical workflow is taking cells, infect them with viruses, and check what happened at the proteome level. So how protein abundance is affected by viral infection, as well as post-translational modification, mainly phosphorylation sites and ubiquitination site, for the view of the landscape of the activation and regulation on signaling pathways. And we like to connect these proteomic data sets with other approaches, omics approaches, transcriptomic, interactomic studies. And the two main questions we ask when we do such a study is first, how viruses affect cellular processes and how human cells respond to the viral infection. Because when you have a virus in the system, the cells and the human are inducing a strong immune response that we can characterize by the induction of proteins. We know that viruses have evolved to counteract this immune response. We can also identify this molecular interplay, and we can identify what cellular machineries are affected or used by the virus to enhance its replication, for example. And in that project, it's mainly about identifying cellular processes, genes that are promoting viral infection or impairing viral infection. And I'm going to mainly focus on viruses. There are viruses, there are a large family of viruses transmitted by insects, mostly mosquitoes and ticks. And you can see on the top left some usual suspects that you may have heard about: Zika virus, yellow fever virus, dengue virus. They are involved in like explosive outbreaks in mainly Africa and Latin America. And in the US, circulates West Nile virus. You have that on the left bottom part, West Nile virus, which is transmitted by mosquitoes, and Hao-Fountain virus transmitted by ticks. And we can categorize two different like group of viruses, the ones that are involved in neurological disorders. That's the case of Zika virus, West Nile virus, and Hao-Fountain virus, for example. That upon infection, they can disseminate in the body, infect the brain, and induce neurological disorders like encephalitis. And on the part, there are viruses involved in visceral disease. That's the case of yellow fever or dengue virus that are involved in more hemorrhagic fever. But all of them start with the mosquitoes and infecting mosquitoes biting human, disseminating the virus within the skin. There is a small viral amplification within the skin, but eventually the virus reaches the bloodstream and then is disseminated in the body. And then, according to the different flavivirus, it can go to the brain, to the liver. The West Nile yellow fever has strong tropism for the liver. But in most cases, for neurotropic flaviviruses, West Nile, Zika, it's mostly asymptomatic. So you have no symptoms, and sometimes some flu-like symptoms. And it's only a minority of cases that could be a severe case of infection that needs hospitalization. And in these cases that are pretty low, it's again very rare to have like some lethality from this infection. And if we want to focus on one of those viruses today, it might be West Nile virus, which is endemic in the US. It's actually been introduced to New York in 1999. And West Nile virus infection is the first cause of viral encephalitis in the US. So since its introduction, since like 25 years, you have around like 60,000 human cases that were detected, 30,000 hospitalization, but this number is widely underestimated because, as I said, few cases are actually leads to hospitalization and infection. So it's more probably like around like 500 million infection we have since 25 years. So every year we have around 1,000, 2,000 hospitalization due to West Nile virus infection. And this is mostly during summer as a seasonal infection. So all viruses have a common molecular biology and a common replication cycle. Like all viruses, they need to reach a cell to replicate. They are intracellular parasites. So viruses bind cellular membrane. They enter by clathrin-mediated endocytosis. They reach the early endosomes and the late endosomes to deliver their genome. They are RNA viruses, meaning that their genome behaves like a messenger RNA that might be translated to express 10 viral proteins. And these viral proteins orchestrate the viral life cycle first by allowing the replication of the genome, so to multiply the genomes and generate more copies. And then the structural protein that are assembled to generate new particles. And our project was from the very beginning a collaborative project. It was a collaboration between the REMEDGE lab, Thomas Jefferson University, and the lab of Noah Duvenet. They were interested in interactomic data between like different viruses, West Nile virus, Powassan, and born encephalitis virus, which is circulating in Europe. And what we realized from these data sets is the viral protein called NS5, the non-structural protein 5, which is pretty important for the virus because that's the protein that's going to amplify the genome and generate new copies. All these NS5 from different viruses interacted with USB7. And when you have such a conserved interaction, it might be likely connected to a function that could be helping the virus to infect the cells or promoting the viral infection by promoting nativity. So that was the first question we wanted to ask: what could be the function of USB7 in viral infection with these flavors? And what we identified is that when we deplete USB7, we have an increase of viral infection. You can see here on the top right, we use sRNA to almost totally deplete USB7 expression. And we infect cells. There are F509 cells, there are epithelial cells that we can follow. We infected them with Zika virus on the left side, West Nile virus on the right side. And in the abscess, we use like an increasing amount of virus. So you have like more viral infection, the more virus we put basically in the control. We quantify the infection by using like FACS analysis. We quantify the number of cells that express a viral protein, so we get the percentage of cells infected. And basically what we observe is in the control condition, the more virus we put, the more infection we have, until reaching 100%. And with the depletion of USB7, we get 10 times more infection, which is something that starts to be important to look at. And we confirmed that this infection was promoted first on the left side of the experiment I show you at the protein level. Then on the RNA level, we can quantify the whole RNA by infecting them for 24 hours and extract the total RNA and quantify specifically the virus RNA. And again, we observe like 10 times more viral RNA without USB7. And on the right side, this is a quantification of infectious particle. So after the infection, what particular viruses are released. And the same situation, 10 times more viral particles produced upon depletion of USB7. So this shows some sort of antiviral activity of USB7 all across the cycle of the virus that promotes both infection and the production of new particles. And something of interest for us, in the lab we have introduced CRISPR-Cas9 base editing. But this is a CRISPR-Cas9 technology that uses the Cas9 and an editor to introduce point mutation in the genome of our cells. So we have an editor, we can have citidine deaminase that introduces C to T mutation, or deaminase that introduces A to G mutation. And working on the USPs is quite convenient because the activity relies on a catalytic core that involves several amino acids that we can mutate each of them and expect to have a loss of function mutation. That's what we did for USP7. We generated like 3 mutants, and here you get the percentage of editing efficiency in a population. It's between 20-40% editing. And again, when we infected those cells, for the control are empty single guide RNA, there are cells expressing non-targeting guide RNA that we infected. We reach around like 5 to 20% infection. And when we introduce this point mutation, there are not the mutation found in patients. There are mutations that we know should be loss of function, like strong loss of function mutation. It promotes flavorless infection. This is a Western infection by 2 or 3 times, as we observed with the depletion using sRNA. So first conclusion, there are some flaviviruses that circulate in the US involved in neurological disorders. And we have found that USP7 somehow regulates the infection of these flaviviruses. Something I didn't show you, but we can discuss, is we have shown and determined that it was independent on interferon system and innate immunity. So that's really an intrinsic activity of USP7. It doesn't rely on a problem of innate immunity and a broad antibody response. So then we wonder how USP7 could have this antibody activity. And if we go back to our replication cycle, we have the tool, a different way to understand where USP7 is acting. If we look at the very early stages of infection, the very first time points, we know that we are looking at viral entry, so this is before any sort of viral replication. Then if it's not affected, we know that we are looking at viral replication that happens in the endoplasmic reticulum, where multiple copies are generated to amplify the virus. And after that, we're looking at late stages that includes assembly of the virus and production of new particles. And then to understand where USP7 was acting, to make a long story short, what we realized is when we infect the cells, we quantify viral infection at very early time points, after 1 to 2 hours, we already get 10 times more infection. And this is by quantifying viral RNA. So we are really looking at the early stages of infection when the virus reaches the early endosomes or the late endosomes, because after that, before that, at that time point, we don't have viral replication that starts. So it's incoming particles that enters more easily within the cells. And thinking about USP7, endosomal entry and viral entry, we went to this key publication, of course, linking USP7 to TRIM27 and its role in the activation and regulation of the WASH complex and endosomal recycling. So we wanted to understand if USP7 function was connected to this complex. So we silence on the top right TRIM27 and a subunit of the retromer, BPS35, and see the consequence of the infection. And what we realized is the silencing of TRIM27 on the left side in orange had the same effect that the silencing of USP7. When we silence both of them and we infect the cells, we get 10 times more infection than in control. But it wasn't the case for BPS35. So it seems to be retromer independent, but it seems to look like something related to the complex of between USP7 and TRIM27 in that regard. So we confirmed that TRIM27 had the same antiviral activity against West Nile and Zika virus, and the same phenotype was observed. We confirmed that in our cell lines, when we deplete USP7 on the left side, we also deregulate the expression of TRIM27. So we are looking at the interconnection between the two. And we confirmed that TRIM27 silencing had the same effect on ball entry and has a strong, promotes strongly ball entry. So we are pretty sure we are looking at the complex of USP7 and TRIM27 together that somehow regulate ball uptake on the endocytosis of the virus. So it might be something indirect, just like that's the consequence of just about here at this stage of the infection. If we stay in the hypothesis of USP7 and TRIM27 involved in endosomal recycling, there is a possibility that the virus actually enter within the cells and get released back by autosomal recycling. That's something which is not very well known in the field in biology. So we have all interest to study that and better understand this phenotype. And we can hypothesize that when we deplete USP7 or 227, we push the virus toward what it wants to go to the late endosome to relieve its genome. But again, so far we need like more data to connect all of them, and we are not sure that the virus is actually itself going through the autosomal recycling trafficking, or is it indirect. But we are pretty sure it involves autosomal recycling, because when we silence and we target genes, canonical genes involved in autosomal recycling and autosomal trafficking, sorting next in 27 involved in the retromer have no effect on the left side of the infection. And then we targeted different RABs. There are GTPases involved to orchestrate autosomal trafficking. RAB4 is involved in fast recycling, RAB35 and 11 in slow recycling and exocytosis. And when we silence those guys, RAB11 and 35, we promote again the infection by 10 times, so very close to what we have in 327. So we still have many things to connect together, but we are pretty sure that this is an activity between USP7 and 327 regulating ball entry. And it's likely to be independent of the retromer, but involved in some way autosomal recycling to promote ball infection. And has a future experiment, and that's something I suggested to get funded by the uplifting athlete with the young investigator lab. I wanted to thank you guys to send me again this email to show me that it was an opportunity. So what I suggested to do to continue was to use base editing to introduce the actual mutation from the patient, the pathogenic variant of USP7, to study its role on flavirus infection. Make a list of viruses of interest by screening different viruses that we know they enter by autosomal entry, to get the first list of virus work to have some surveillance on it and to understand the spectrum of activity of USP7. Use this fibroblast from combined brain and biobank to then see if we recapitulate what we see from model cell line primary cells and infect them with flaviruses to see the effect. So all of that to build some preliminary data and then get all the funding to do some more in vivo studies and really like go to the pathogenesis and the infectious disease to better understand how it would affect, and does it recapitulate what we see in cells? Does it like promote viral infection, but also does it promote the actual disease, the infectious disease? So thank you, everyone. Thank you to the different labs that collaborate with us. Thank you to Combined Brain for this very good organization and this exchange. And if you have any question again regarding this virus infection, innate immunity, feel free to reach out. And thank you so much for your attention. If you have any question, just let me know.
Not Mexican, plastic retriever, that's another recycling agency.
Yeah. So the retriever is another complex involved like in autosomal recycling, right? So what I suggest, what I didn't show is like we have some specificity regarding flaviruses. We know it affects Zika virus, West Nile, it's not dengue or yellow fever virus, for example. What I suggested was to screen all the structure, the retriever complex, the scaffold complex that breaches between like late autosomal endosomes, ER, for example, to understand the path of entry of these different viruses, and we will know and we will map the different structures that are involved in this phenotype or that involved in other interaction with the virus. Yep.
Great. Can you remind me how did you find out the connection between the polymerases, I guess?
Yeah. Yeah, so first it was interactomic data sets. We observed an interaction, and NS5 is very well known to hijack cellular factors to benefit the replication. So our first hypothesis was, oh, it might be beneficial to the virus, not antiviral, but beneficial. Maybe the polymerase use USPs to avoid some sort of like replicating tag and degradation. So we started to screen it, but it was the opposite. It was antiviral. And when we look at this staging experiment I showed, it was not related to the NS5 at the end, it wasn't related to the polymerase. We suspect that actually you can have another function during genome replication because we know that NS5 also recruits some membranes to hijack, to like hide itself to avoid innate immunity sensing. So we think there is another aspect regarding NS5 USP7 interaction. But we are pretty sure that the bulk of our phenotype, of our antiviral phenotype, is dependent on entry.
Well, thank you. I really, really beautiful plans that you showed here. I would be interested to hear from the families whether there is experience with kind of increased susceptibility towards viral infections of that kind. In particular, you know, as it relates to the neurotrophic viruses that you presented, which potentially could be concerning in individuals who already have a neurodevelopmental disorder, and who could have like quite a compounding effect of experiencing infection with such viruses like West Nile, you know, which is very common.
Yeah, exactly. So that's my question for you all. If you ever experience like severe infectious diseases, or you think about that, it might happen early after birth to have like repetitive and recurrent infection diseases. I know some of the kids and some individuals are IgA deficient, and so I know that some of them get recurrent infection, but it doesn't seem to be like severe cases that needs hospitalization and help from that. So on respiratory virus, I think if any of you didn't experience like these severe cases very early, it might not be connected to respiratory viruses. But the case of West Nile neurotrophic plague viruses, that's something, because it is present, it's not that well detected, it's not always screened, and I don't know what could be the confusion between the encephalitis or neurological disorder, which is sort of like expect from the genetic disease and not from viral inputs. But yeah, that's something we want to push forward to understand, like, is it a risk factor? And again, what I show you is like only on cell lines, but like the actual infectious disease and what happened in the body is complex of several levels of interaction between immunity and the virus. So that's a totally different approach and like question that I show you, and mainly uses like in vivo models, for example.
I think it's quite important to keep that in mind. I mean, fortunately, I'm not aware of like many episodes of regression amongst individuals with Hao-Fountain syndrome. But if there was someone who experienced unexpected neurological regression, I think it's important to keep that in mind, because like is your analysis of like West Nile and such viruses will not be part of the standard workup that would be applied to giving you data. I think that should be happening.
Yeah, exactly. That's what I would like to implement, and to be interaction with the crew to be sure that we can do that. And yeah, exactly. And that's why it's so interesting actually to have this first data, because what we expect to do from that actually generalize more to other genetic diseases, to be sure that we are not like facing this kind of interaction for different neurological disorders. And we have different aspects in that, study the virus in the patients, right? And the consequence of the infection or the immune reaction on the disease. And that's something I want to pursue. That's why I like asking all the grant to, as I told you, like grow my lab to study specifically that the interaction between viral infection that can happen anyway, and genetic infectious disease, because that's something which is not studied, not known, because there is not yet this connection. But actually, West Nile are already being connected to some like neurological disorders, Parkinson, Alzheimer's disease, for example. So that's the sort of interaction we want to study. Thank you for raising that. Thanks. Again, any sort of question after in this case.
Give me my glasses. See you. Oh, what percentage? This is big.
I can have ice cream. Salad. Right. Mommy. No. Yes. No. Yes.
Okay, next I'd like to introduce Lisa Ladd for our next presenter.
Yes, thank you all for having me here. My name is Lieselot van der Laan, and I'm a PhD student from the Human Genetics Department, Epigenetics in the Amsterdam University of the Netherlands. My PhD mainly focuses on DNA methylation in rare genetic disorders, and one of the disorders that we are studying is Hao-Fountain Syndrome. And today I'm going to tell more about the DNA methylation episignature in comparative epigenomic profiling of Hao-Fountain Syndrome. Maybe if you remember a few years ago, I gave an online presentation where I was asking for participants for this study, and today I'm going to show the results. So for some terms to make it easier for their understanding of my presentation, let's start with DNA methylation. DNA methylation are chemical tags, most of the time metal groups, on top of the DNA that can be switched on or off. This depending on where they are in the genome, and this can also regulate whether genes are turned on or off. Episignatures are unique DNA methylation profiles for specific disorders, and those are also cell-specific, so meaning that skin episignatures can be totally different than blood signatures. And at the moment, there are more than 100 episignatures, for example, for well-known syndromes like Down syndrome, and also for rare syndromes, imprinting disorders, and for environmental syndromes, for example, fetal alcohol spectrum disorder, which is caused not by a pathogenic variant in a gene, but by a mother drinking alcohol during pregnancy. And all of those episignatures are included in a diagnostic tool called EpiSign. The EpiSign is launched by Bekim Sadikovic in Canada in 2019. And at the moment, medical doctors are using EpiSign to diagnose patients and to reclassify variants of uncertain significance. So we know that genes with epigenetic functions are part of the epigenetic machinery. And those genes can be divided based on their function, if they are eraser, reader, writer, or remodeler. And we know that if there is a pathogenic variant in one of these genes, it can influence the DNA methylation and therefore cause an episignature. So if you look at the function of USP7, it's also a gene that belongs to the epigenetic machinery. We all know that pathogenic variants are causing Hao-Fountain syndrome, and that the gene is located on chromosome 16p 13.2. If you look at the function of the gene, it's a chromatin remodeler. It controls the DNA packaging and gene activity. It's also a deubiquitination enzyme, which means that it removes ubiquitin from proteins. And it also regulates the MDM2P35 pathway, which is an important pathway for cell growth. It also has roles in DNA repair, transcription, and cancer-related processes. And based on those functions, we hypothesize that it would be possible to generate an episignature for this gene, which can help patients for diagnosis and reclassification. So therefore, in close collaboration with the Heidelberg group, especially with Professor Schaaf, we included a total patient cohort of 18 individuals. We started with a discovery cohort, which meaning that we discovered the episignature and trained the episignature with a total of 12 patients. Later, we found out that 12 of them, 3 of the 12 were not responding the same as the others, so we call them outliers. And I will tell later more about why and how. We also added a validation cohort with 3 pathogenic cases and 3 FUSs to test and validate the episignature. We also used some controls that were matched on age, gender, and VATS to see what the differences were between the patients and the controls. So in A, you can see one of the patients that has a large deletion, which not only included USP7, but also other genes. And B is showing all the point mutations. So most of the patients have missense variants. We have some with frame shift and splice site. So a short part of our study was the clinical characteristics. And out of the 18 individuals, all of them had intellectual disability, developmental delay. Most of them had speech delay, behavior abnormalities, and hypotonia. And here you can also see the photos of patients that were included in our study. And you probably will see a few that you recognize. Some less frequent features in our cohort were epilepsy and microcephaly. So back to the episignature part. After that, after we included the patient and we got blood, we isolated DNA out of it. And we did run the EPIC array, which is an array consisting of 850,000 CpG sites, also the chemical tags, that will be measured on methylation. When we got the data back, we performed a quality check. In R, we are using our bioinformatical tool to analyze all the data. We then performed some different methylated probes and classifications for vector machine analysis. And those are helping us to see if there was a difference in methylation compared to patients and controls and compared to other episignatures. And we finally performed some different methylated regional analysis where we looked into DMRs, which are regions of the chemical tags in direct order of each other. And those can tell us something about other genes where USP7 is working together with. So if we go to the results, in A, we see our heat map, which is showing a big bar starting from red to blue. In red, we have our patients, and in blue, we have the controls. And below, you can see a lot of colors from blue to white to red. Blue meaning hypomethylation, so less methylated compared to controls, and red is meaning more methylation compared to controls, so hypermethylation. And you can see that our controls and our patients are separately clustering, meaning that they show a totally different episignature methylation profile. And if we make this a bit more visible, you can see B, our multidimensional scaling plot, which is showing us again our patients in red clustering separately on the controls in blue. If you look closer, you can also see some purple dots clustering with our patients. That's our validation control, the validation cohort, which are acting the same as the patients. And you can also see that there are 3 orange dots clustering with controls. Those are the so-called outliers I spoke about before. And I will go back to that later, what might be the reason they are behaving differently. So we do have an episignature for USP7, but we also want to know if it's reproducible and robust. So for that, we are using a leave one out cross-validation, which is basically removing one patient every round and add it later in as testing. And every time the tested patient clustered together with the other patient, meaning that we have a robust and reproducible episignature for USP7. But then there's one thing we would like to know, and that is if it's unique and if it's not overlapping with other episignatures for other disorders. So therefore, we are performing a support vector machine classifier. And below you can see a lot of names. Those are all syndromes that were part of the EpiSign tool at the moment of the discovery of the USP7 one. And on the other side, you can see the MVP score from 0 to 1. 0 meaning absolutely no overlap, and 1 is meaning a total overlap. So you can see all the blue dots. Those are patients with specific disorders. All of them are staying at 0, meaning there's no overlap at all between our USP7 episignature and all other disorders. And if you totally go to the left, you see a lot of blue dots at 1, and those are showing us our USP7 patients. So logically, they are all at 1, showing USP7 signature. So let's get back to the so-called outliers or patients with a negative episignature. Those are the 3 patients. And it's firstly good to know that negative episignatures are not meaning a negative diagnosis for this syndrome. It's just meaning that they behave differently than the other patients. And there might be a few reasons. It's possible that those patients have a more severe clinical future, or that a variant is in a different domain of the gene. We saw this with a lot of episignatures, that when the cohort is relatively small, that some of them are behaving differently and clustering with controls. So it also might be possible that in a few years, when the cohort expands, that those patients are again clustering with the patients. So I noted the 3 outliers with an arrow, and as you can see, they are not really in a different domain together. There are other variants in those same domains that do respond on the episignature. So we are not sure why, and we hope that maybe in a few years we can have a bigger cohort and see if they do behave the same as the other ones. But again, good to know those patients do have Hao-Fountain syndrome based on clinical and molecular features, but they do not have the same episignature. So we also performed some global DNA methylation profiling, and this is showing us that the episignature of USP7 is one of the most hypermethylated episignatures on the EpiSign tool. And if we look into the DMRs, the different methylated regions, we found 35 of them, and most of them were in promoter regions, which is causing genes to turn off. And all the rest of them most were in intergenic regions, which block enhancers, and those can also misregulate genes. So if we look closer into those 35 DMRs, we found that 4 of them were of particularly interested, because those are also genes that are annotated to neurodevelopmental and intellectual disability. My colleague Manassa will tell later after my presentation more about DMRs and how this can be linked to gene expression and etc. So more about it by Manassa. To conclude, we identified the highly specific episignature for USP7, and that episignature is already used in the EpiSign diagnostic tool. It already diagnosed around 6 or 7 patients that were previously undiagnosed, and we hope in the future it can help to reclassification of USP7 variants and enhances clinical decision making. If you want to know a lot more about EpiSign, please find me after the presentation. If you, for example, have patients with FUSs that you would like to have tested, or you want your own child to have tested for the episignature, please reach out to me. In Europe, the Amsterdam, the lab where I'm working, is doing the EpiSign analysis, and in the States, we have the Greenwood Genetic Center that you can also ask for that. So researchers never performed alone, and therefore I would like to thank a lot of people, especially the patients, because without you it was not possible to make this episignature and to help future patients, especially the Foundation for Hao-Fountain Syndrome to grant me with a travel grant, which made it possible to be here. The Heidelberg group, especially Christian Schaaf, Sebastian Saylor, and Pilar Bureau for helping with the inclusion of all the patients. The clinical genetics department of our lab, the lab from Beckham Sedikovic, who invited me for 3 months in Canada to work on this episignature myself, which was really helpful. And my own lab, especially my supervisor, Peter Henneman, and my dear colleague, Manassa. So that was it. Thank you. Oh.
No, you guys found some episignatures associated with other disorders. If you know, we talk about looking at your information.
So for other disorders, you mean?
Yeah.
In relation to USP7 or?
No, just like if you found an episignature that goes with another disease, what are what are they doing with that?
Like that. Yeah. So most of the time, especially in our lab, what I hear from the specialists, it sometimes can be really difficult to diagnose a patient Most of them have overlapping features like developmental delay, speech delay, and they perform whole exome sequencing most of the time. But that's really expensive. It's 3 times more expensive than EpiSign. So for nowadays in our lab, most of the times they ask for an EpiSign first. Then they found like the episignature, for example, USP7 or Down syndrome or any other. And then they perform a targeted sequencing with Sanger. So it makes it cheaper and they can have more insights. Yeah, so that's mostly why people use it nowadays.
I would say like another kind of meaningful situation could be that an individual with a neurodevelopmental disorder gets exome or genome sequencing, and there are multiple variants of uncertain significance kind of identified, several of which might be de novo or not be carried by the parents. So like even after genome sequencing, it could be uncertain as to like which one of those variants is actually causing the disease and like what is the actual diagnosis for this patient. So here the episignature can also help. Let's say we have the variant in USP7 and the variant ABC12, and we do the EpiSign test, and that may determine quite specifically, well, the episignature is representative of what we see in Hao-Fountain syndrome, and that may then confirm the diagnosis. On the other hand, as you see, every individual who has been involved with Hao-Fountain syndrome falls into the episignature, so there may still be kind of like either secondary signatures or kind of other explanations.
Yeah, exactly. And the good thing is that EpiSign every year runs again all the patients that did not have a diagnosis. So it might be possible that, for example, if you submitted the patient a few years ago and the episignature of USP7 was not there yet, it will be run again against the new signature. So if you submit a patient, it might be possible that in 5 years you will get a diagnosis of a new one. Yeah, it's really useful.
Thank you for wonderful presentation. Don't have the time to do it. I have a question. Is there any information about Prader-Willi syndrome patient and shutdown? The syndrome patients on the episignature?
Yeah, so that's a nice question. We're working on the Schaafjong episignature at the moment. So we hope that at the end of the year it might be possible. For Prader-Willi, I thought there is one. So if there are FUSs in this syndrome, they can be sent in for diagnostic.
Would be interesting to see any overlap in the figures in shutdown patients.
Yeah, especially that's why we started this with Professor Schaaf as well, to see if there is an overlap in episignature. Should be if we look at the functions and the working together of the genes.
Just a comment that it's curious that the 3 outliers that you found, 2 of them we find that they're pretty positive.
Yeah, I think we spoke about it with Bo and Professor Schaaf as well. So yeah, thank you.
So just to follow up, what does it mean? The they were normal? Yeah. They don't cause really in vitro. In vitro.
Okay, next I'd like to introduce Manasa, and I would pronounce her name, but I would butcher it. No. You have Bruce as well.
Hello, everyone. I'm Manasa. I'm currently working as assistant professor. Just started with research in USP7 just a couple of months ago. I joined team with Liz Lott. We are from section epigenetics, Department of Human Genetics, Amsterdam UMC. So whatever Liz Lott has presented until now about the epi signature, we're trying to take it a little forward in terms of understanding the functional relevance of epi signatures, but in our own way, in a new way, I would say. So the title of the presentation is Decoding USP7 Dysfunction. Also to mention that I represent here from Amsterdam UMC called Emma Center for Personalized Medicine. This is a newly formed expert group, a consortia well within Amsterdam UMC to bring clinicians and scientists together to focus or to fast track research and therapy just for the rare diseases. So in that name, there's a lot of database, biobanking, and patient information that is coming out. So yeah, that's also a great initiative. And yeah, through that, I'm contributing towards USP7 research. So to give a very brief background of what I'm trying to do, brain development in humans is achieved through very precise spatial temporal genetic control, mechanisms of which is largely not known. To be very specific, it's not a uniform process. It's tightly orchestrated sequence of events, starting from fertilization. There is formation of 2 cells, then it forms 4 cells, 8 cells, 16 cells, and then it already hits the phase of a neural tube formation. Neural tube is a highly important critical window of development. I would say this is a critical window for great cellular expansion and cellular diversity that we see. In common terms, can say as blueprint for the development of the entire human body later on. There is already formation of neural tube as early as post-conception week 4 to 6. And this phase of the brain development or the preprint, it is called as neurulation. And very quickly it hits neurogenesis, which is marked by the very much like the proliferation of neural related progenitors, neurogenesis. And then already through the mid-gestation week, we see already the differentiation, neuronal migration, synaptogenesis that will all start getting kicked off from the mid-gestation. And then already through the postnatal development until the adulthood, we still see myelination and still see the synapse refinement. This is the overall biology of development or the status of brain development. And research till date has linked up 2,500 genes to neurodevelopmental disorders. This clearly points towards how complex the genetic landscape is. It is a very good review paper from 2024 that tries to map all the known brain developmental disorders onto this spectrum of biology of neurodevelopmental stages, starting from neurulation, neurogenesis till oligogenesis, synapse development, and myelination. As you can see, a couple of disorders that you probably might be familiar of, for example, Hao-Fountain Syndrome. There is something wrong with the neurogenesis state, which is very much early. And then there is, for example, in synapse and circuitry, there is problem with, like, for example, Fragile X syndrome falls into this category, Rett syndrome falls into this category, many more examples. So molecular evidence to place Hao-Fountain Syndrome on this entire spectrum is limited. I would actually say it as there were no evidence until recently, a month ago. In 2025, Shen et al. From Washington Medical University, they have published the first ever evidence connecting Hao-Fountain Syndrome to the actual molecular biology or the neurobiology of the disease, connecting it to something wrong with respect to the synaptic development and morphogenesis. This is the first ever molecular evidence that talks about what could be going wrong in terms of Hao-Fountain Syndrome patient neuronal models. So apart from this, there are no other evidences. It is extremely important to position USP7 disorder within this framework, as this helps very clearly to map on which developmental processes are most vulnerable for USP7 dysfunction. It guides therapeutic strategies by linking molecular pathology to developmental timing, as well as to the neuronal circuitry. So with this context, I would like to introduce the guiding questions of a study to understand what could be going wrong, what is the molecular mechanism of USP7 dysfunction. So whenever a risk factor for neurodevelopmental disorder is identified, there are important 3 questions that we can ask. How does this risk of mutation or USP7 variation can transform itself? A genetic risk can transform itself into a perturbation that leads to the consequences that we see later on. The second question is where during the development do these perturbations occur? And question number 3 is when during the development are these perturbations most critical? And as you can see in the picture, it depicts anatomical correspondence between the developing brain regions in the early embryonic brain development towards the postnatal brain. And as you can see, it is already decided what is going to become hippocampus, what's the midbrain, what's the hindbrain, what is going to become what. So there is already a layout that is going to already, there will be a blueprint already existing. So to address this whole question, we had to come up with a framework to answer this question on how a risk is transforming itself into a perturbation. So combining our expertise and the existing literature, again, pointing towards the one of the original publication that has inspired a couple of researchers like us is B. Johnson's publication from 2015. This is also what Lee Slot showed in the previous presentation. This is a picture. As you can see in the final layer of the plot, you see the names of the genes. I have also marked some of them. There is a mutation in this gene that has been associated to a disorder, and the disorder is basically a Mendelian disorder that is considered. So, for example, DNMT1 is associated, for example, to a disorder, Sotos-like syndrome, or the ICF syndrome, as you can see. I'm sorry, the plot is not very clear. So each one of them, each mutation is associated to a Mendelian disorder, and they are belonging to a category of chromatin binders like epigenetic machinery. They are capable of influencing the disorders that we see later on. What is the take-home message of this entire plot from this original paper is that all these diseases which are associated with something wrong with the chromatin machinery had one very well-pronounced clinical phenotype, and that's intellectual disability, which is shown in the yellow. And there are also others like immune dysfunction and recurrent infections shown in purple color, for example. So this clearly says that the intellectual disability is kind of very well pronounced in case of epigenetic machinery dysfunction in Mendelian disorders. Other way around is also that the clinical manifestations that we see much later during the development can actually be an echo of early molecular disruption that we are seeing. So this is the hypothesis. And to support this also, USP7 has higher expression or is already active as early as 8 cell state. And it is also one of the important genes that gets transcribed when the zygote starts reading its own genome. This is called zygote genome activation phase. And here already one of the 800 genes that get transcribed from the zygote in itself, USP7 is one among them. So can be pointing towards an echo of early disruption, like I said. And to see this all and to understand this in real, what we used is patient fibroblasts as a window to understand USP7 dysfunction. And for this, why fibroblasts? I think this is quite an established question currently, thankfully. Patient fibroblasts, they still retain donor-specific molecular signatures. There are enough publications that have confirmed this. There is a success story about Rett syndrome in itself, where patient fibroblasts were used to reprogram them to neuronal models that proved that the epigenetic and the genetic and the epigenetic signatures, they retain the disease signatures along with them, also used for key mechanistic as well as therapeutic insights. They used IGF as a therapeutic molecule, and they tried to prove that the neuronal models can still, they can serve as a therapeutic study models to prove therapy in Rett syndrome. So what we use is fibroblast cohort coming from 4 patients and 4 controls, and all the 4 patients had pathogenic USP7 variants with 1 deletion. And then the controls, important thing is that we also considered that all the 4 patients are also epi signature validated, like what Lee Slot just mentioned. So all the signatures identified in the patient blood, we also considered the cohort so that they are systematically connected. So with this, we go forward with our approach. The approach was to use the multi-omic insight. Like I said, one was to study about the epigenetic machinery, and the output of that is, of course, the gene expression, like the transcriptomics. So we are going to use step by step, trying to build on the hypothesis of how can Hao-Fountain syndrome can be mechanistically studied using multiomics. So the first part is to start with the transcriptomic alterations. So when the patient fibroblasts are compared with the control fibroblasts at very classical differential gene expression, it yields 310 differentially expressed genes with many upregulated and many downregulated. They clearly point towards positive neuronal regulation in terms of enrichment. They also point towards regionalization as well as peptide assembly. So in clear, the USP7 dysfunction also points towards that there is mechanisms underlying neurological manifestations. Some of them also have quite literature associations in terms of intellectual disability or autism spectrum disorder in general. And the next was the DNA methylation that we topped it up because that's our current expertise. So we also did the methylomic understanding. We used a methylation array to do this. And in terms of this, we identify nearly 1,000 genes, genomic regions that are differentially methylated. The overall trend is also we observe more hypermethylation in patients compared to the hypomethylation. This is also a similar trend that we see and have reported in the blood, for example. But why it happens is something that we still have to explain. And it is possible to go forward from methylomic alterations to connect them towards the gene expression, which we do in the coming slides with a preliminary analysis. So in terms of overlap of the differentially methylated regions between patient blood or the episignature that is presented before and to the patient fibroblast, it's very clearly not many overlaps. We see only 8 regions that overlap between patient blood and patient fibroblasts. But on the other hand, it's also important to note that the blood is quite a bulk tissue, and we still have to go much in depth to compare it to a cell type like a fibroblast. So in any basic association between DNA methylation epigenetics towards gene expression start with this basic classic model, especially the DNA methylation. So if there is a promoter of the gene, which is just starting from the transcription start site to the 2000 base pair substring, it is called as a promoter. Promoter has to have a transcription factor come and sit on it so that the transcription gets initiated. So if the promoter has more methylated states, like addition of methyl group on the promoters, it is not possible for transcription factor to come and bind to that segment of the DNA and initiate gene expression. And gene expression is extremely important. Like I said, the whole neurodevelopmental disorder can be spatiotemporal genetic control. So in that sense, gene expression is something that plays quite a good readout for us to understand if something is normal or not normal. So this is a classical model where we just see the first 2000 base pairs and try to associate that the differential methylation is associated to a gene expression. But it's also possible to extend this model much beyond. This is to move towards an enhancer. Enhancer is any other genomic region or CPG island region that has capacity to influence the gene expression sitting far away. So when we say far away, what would be the distance, right? So in terms of the existing databases or very well studied databases in epigenetics, the ideal distance or the maximum distance that a gene can get influenced from a farther enhancer molecule is 250 kilobase pairs. So I have also considered 250 kilobase pair to associate differentially methylated region to influencing a gene expression. But this is an assumption, and we have to go forward with the assumption to see how it looks for the next steps. So with the 250 kilobase pair distance, when I try to associate gene to a differentially methylated region, at this point of time, I have to say computationally as putative gene enhancer pairs, meaning they have to be validated later on in the wet lab. So they're still putative. And this is a very simple statistics to start with. This is correlation, just a very basic correlation between differential methylation and gene expression with the assumption that differential methylation influences expression. So what we clearly see is the Hox cluster, especially the Hox-B, is harboring many differentially methylated regions around it in the distance of its 0 kilobase pairs to 250 kilobase pair distance. So out of the 10 differential methylated regions that sits around it, 6 of them share a statistically significant correlation between gene expression and differential methylation. So this is the starting point for us. Out of those 20 different targets that we are reporting in this research, I'm just presenting about Hawkspeak Award for the confusions or to keep it well within the time. So Hawkspeak cluster is represented with 6 significant associations to DMR, to differentially methylated regions. 5 of them are positively, negatively correlated, and 1 is positively correlated. What does that mean? It's like a typical, if there is increased differential methylation, then there is reduced gene expression. This is according to 5 different differential methylation regions. But the 1 differentially methylated region that sits right on the promoter of this gene is actually positively correlated in terms of the correlation analysis. But we do have to go forward to prove them as functional relevance. So this entire work, along with what we have reported in the blood and what we have reported in the fibroblasts, including the cross-tissue comparison, is all now sitting as a manuscript and is under the review. What's the next step for us is, like I said, we have to go for the functional relevance. The plan to go forward with the functional relevance is like this. There are already existing databases that do talk about whether the genomic region, which I'm calling as differentially methylated region, is an important region or not based on its activity or availability in terms of the H3K27 trimethylation, acetylation. We have so many other possibilities that the genomic region still can be active, can be sensitive. We are going to use all of those existing data sets. Number 2, it is also, there are many databases that are coming up with huge information on fetal development and embryonic development. So we will be able to understand if this specific genomic region had any relevance or sensitivity during the developmental window. That's the second thing. So this is all the bioinformatic pipeline, but also parallelly, we do have our own experiments ongoing, which we are yet to finalize, on explaining that if the hypermethylation is having any influence, the hypermethylation that we see in terms of the patients compared to controls, do they have any information in terms of the permanent silencing of some critical genes involved in neurodevelopment? That's the hypothesis. So with this, we are able to, we are very hopeful that we will be able to conclude from these experiments that there is a molecular perturbation of FUS7 drives neurodevelopmental disorders by active silence, which is critical for lineage specification or something else. So we are on almost like 70% there in building this experiment and hypothesis. We'll keep you posted once there are some solid results from this research. So with this, what we are hoping is the objective 1 to address how genetic risk transforms itself into a perturbation. The first mechanistic insight on how the mechanism is all driven, we will be able to answer. That's the current status of the work. The next status is to answer where and when. For this, we are going to utilize the iPSCs derived from patient blood that we have to understand exactly when and where something is going wrong in terms of USP7. So this is it in terms of the work. So would like to acknowledge all the people who have contributed, and I think most of you also I'm personally meeting for the first time. So yeah, thank you one and all, and especially thanks to the patients and the family. And I will not. I'm quite moved by the introduction that I heard from Mangan and Bo. Yeah, that was really great, and yeah, it's good to keep us in the reality of why we do this research. Thank you very much. And for any questions or comments, feel free to reach out. Thanks.
So we were going to segue to Dr. Fontaser kind of hosting an open session on the presentations that we just saw. So I feel like this would be a great time to get that started. And then after that, we'll have a break. So if there's questions or if you want to discuss like everything that we've seen or that kind of stuff. Do you want the microphone? Okay. I know this is a new format for us that we're kind of having these little mini discussions afterwards. I think it's on.
Do you hear me?
Yes.
First of all, I have to apologize to be late. It's really crazy. I come from Amarillo, Texas, desert. We almost don't have rain. I couldn't come because of thunderstorm was there for 3 hours, and I missed my connection in Dallas. But I'm very glad to be here with you. I'm honored to be participating. I think it's really, as mentioned before, these efforts that are driven by parents and clinicians and inspire them also to bring fundamental basic scientists in to really help move the field forward. So with this, I would like to open the forum and invite all of you to address questions to all of the speakers from this first session. I think that's the point. And I have a couple of students, Tara, Bayat, and Denish Tepiha from my lab. They will take notes. So Amber also let us know we need to have notes. So we'll do that as well. So please, the floor is yours. And maybe if you don't mind to introduce yourself and where are you from, so we will know each other better after this session.
My name is Jennifer McCollum. I'm probably loud enough. I'm Jennifer McCollum. I live in Dallas. My daughter is Madeline. She's 8. She is one of the outliers on the episignature. And so I'm just curious, like, what are some of the reasons? I know we don't know the exact reasons for each of them, but like, what are some of the things that could cause someone to not have the same episignature? And then the second is a statement, which is we have cord blood and tissue. And so I feel like this is the good audience to tell we have that. If you want it, we have it banked.
Yeah, wonderful. I think it's a very good question. So
Thanks for the question. And yeah, there might be different reasons and we do not know exactly. But in different other disorders, we saw that some of the patients had a more severe clinical features or really mild in comparison with most of the patients where the episignature was generated on. So that's one of the reasons. The other reason can be that the variant is in a different domain that can cause also a different methylation profile. Yeah, and that the cohort is too small. That's also one of the possible reasons that, for example, it consists of 22 now positive patients, and it might be possible that, for example, if there'll be 30, that the episignature can also be like wider, and therefore the outliers can be included again. Yeah, so we're not sure, but that could be a few of the reasons. I hope I answered the question.
I have certain follow-up. Well, I'm Emily Gilbert. I'm a faculty at University of Kentucky. Some of these variants fall into the active variants, and some of the variants fall into the inactive, which we will hear later on today a lot about. Can that be a reason you see different signature? Also, USP7 is such a pleiotropic, function-wise protein. Can that be a reason for your differences in signature and really not just, you know what I mean?
Yeah, that could also, yeah, that could also be a reason. Yeah, we're not sure about it yet. And yeah, the team of Beckham is also doing a lot of research about this in Canada and trying to regenerate episignature again and again if more are added. So yeah, it could be a reason. Yeah, simply we just really don't know the exact reason.
Thanks. I also want to follow up because I think it's really interesting. Do you think the episignature could be different depending on what type of cells you're looking at as well?
Yeah, that's what I told as well in the introduction. So we also noticed that in our fibroblast patients, that the episignature is totally different than the blood episignature of USP7.
Yeah, and I wonder if you look at neurons, for example, do you see something that's includes all the patients?
Yeah, we do very clearly understand different episignature indications. And now that we are looking at the fibroblasts, we tend to see fibroblast specific changes in patients. And when we go to the other cell types, then possibly we see the influence of that specific cell type and the microenvironment of its cell number.
that would be influencing the condition. So, like I said, when we try to do the cross issue between blood and fibroblasts, right now we see only 7 to 8 overlapping regions. And yeah, I actually cannot take it like it just because hypothesis if we get it too deep on why only these 7 are overlapping, because we know blood is quite a massive issue to consider what would be influencing the epigenetics of blood, for example. In that sense, fibroblasts would be less known here. Maybe if we go for more differentiated cells like neurons, we could probably see much more clearer information solutions. But without anything that's tissue specific, it's not a great overlap. It's what you expect.
And I also wonder, you know, does things like age or where people live, all these things affect that as well?
Absolutely. So that was one of the first big discussions that we were having before we even start the work, because we cannot ask for the we cannot have age match controls, age match patients also, like, because it has to be kept anonymized, and we had to go forward to like request them please for age information. We don't want anything else. And just the gender and the age, you have to consider all this possible things that could influence our analysis and observation grading. So fibroblast in age is quite an important factor to consider when we have considered them.
It's also good to add that, for example, in blood epi signatures, we see that if blood is taken in the first year of the baby, the DNA methylation is not like stabilized correctly, and those are always behaving differently. But that was not the case.
I'm sorry to hijack this, but I also wonder if you think of maybe a model going forward where you can just have that on mutation. What would be the best way to do it? Yeah, like doing something again, you know, cell culture, which is right. So you don't have to depend on, you know, these variables where, you know, people live or what they eat or all in whatever. And wondering if you know you kind of
that we can go forward with. So that basically is already in program and it's already in state. So it's ideally, hypothetically, it's possible to take them forward towards building a realistic model that is independent of all these variations and influences, but to be also specific. There is a lot of validation at the level of epigenetics that is still pending also in the IPSCs. So we are still far away from it. It should be possible, but ideally we are still far away from it. Because the IPSCs, we are currently using some specific set of markers to see whether they are fully reprogrammed, etc. But at the level of epigenetics, we still do not know. It's still a mystery box sitting there. So yeah, it's possible, but yeah, right.
For you guys too, specifically Lizlo. Lisa, sorry. Is it a paper together? Because I really would love to see the exact mutations, like the number of residues and all that, and like kind of put it with my data and compare what we can see.
Yeah, of course. The paper's published, so that was to send out.
Okay.
And if you want to have more things that are not in the available, then you can send me an email and I can send you the data. Cool. Thank you.
Hello, my name is Allison O'Neill. I'm a assistant professor at Wesleyan University, and I'm using all of the combined brain patient-derived stem cells with their corrected controls to model the disease. So I have all the cells you need in the freezer. So all the neurons and the neural progenitors. So I am looking at our data and thinking about it, and you know, you lose a lot of the epigenetic signatures when you reprogram. Right, so I wonder about the validity of the model after reprogramming. That makes such a big change. Then also fibroblasts are probably not the best cell type to use that's not necessarily involved in the disease. And the blood is a lot of different cells, right? And do you do any kind of sorting that you only look at one kind of blood type, no, cell type, or is it just blood? Right, so it's both, right? So that's a mixture of tissues, so that's messy.
We are currently studying IBSs every second year to see how they behave differently compared to but yeah, we know, like the data.
Right, I think, I wonder, what's nice about I don't know which IPSCs are using, but what's nice about getting all of them from the same bank, from combined brain, is they're all reprogrammed in the same way, which, you know, as a stem cell researcher, we always say in our grants, like, oh, it doesn't matter, they're reprogrammed in different ways, that doesn't affect the pluripotency, but it could definitely affect epigenetics.
Yeah, we agree with them.
Yeah, right, so I think that's another thing just to keep
Because
in
the
mind.
I
I have no, you know, I don't look at I have no, it would be, you know, just be interesting, right, all scientists, but it'd be so interesting just to know, like, if you reprogram, how does it affect the epigenetics, but it has to, right, it has to make them pluripotent.
Yeah, it at some point it's important to keep in mind that to address the problem of sample size, we will probably be overlooking the places it's having in the labs that the samples come from.
Yeah, we're starting a study where we're watching gene changes over the developmental timeline now that we have a really nice, robust cortical, we use a brain organoid protocol. So I'm happy to share all the troubleshooting we've done to get the cell lines to do what we wanted. Because if you guys do stem cell, every line is different. And so
We don't have something for that, but that's the plan.
yeah, please reach out. I can save you so much time. Please reach out.
Yeah, very nice.
Hi, I'm Belinda from North Carolina. I'm a mom. This is really to the gentleman from Tulane with the circadian rhythm research. Wait, where do you go? Oh, he's over there.
No, I don't think he would.
Yeah. So what if you no longer have any baby teeth and you don't have wisdom teeth? Is there any way to participate in your study?
I mean, we can extract them from regular teeth, but I don't think we've ever done that before. We have done wisdom teeth in the past, but we really want the neural crest, like baby teeth, because that way we can age match. And something about the reprogramming I kind of wanted to add to DPSC, so we don't have to do any type of reprogramming prior to differentiation. They're ready to go out of the box, basically. But I don't know if we've done the liberty, like, let's say somebody loses one in an accident. I don't know if we could do that, but we could try. I think that's the best we can offer.
Other pools of natural improved stem cells that could as well. For your study, you're doing cell types the same, but there's other pools of stem cells in adults and children. Would be more invasive and losing too.
Yeah, I think this is really great discussion and also points to many insights of even USP7 basic function that we don't know. And one is, I think, tissue specificity, right? Not just epigenome, but also other functions, how different cell types, different tissues USP7 might function. I think that a lot of value discovery is still waiting. Any other question?
Hi, thank you to everyone who's presented and everyone to be here. My name's Scott. My wife Brittany and I are here. We're very new to this. Our son Felix is almost 4 and was just diagnosed last week. And serendipitously, we live 20 minutes north of here in Nashville. And so when we found out this was going on, we immediately we have to be there. So I know, what are the odds? But as part of our, newness to this whole, we're just trying to get our head around everything. How does we know that the size of the cohort is very small. I think on the foundation website, they listed 250 something identified cases. In terms of the trajectory, we know that this variant was identified very recently. How does this compare to the trajectory of other genetic syndromes that have been identified? So is it expected that the cohort will grow perhaps exponentially as more and more people are tested now that we know that this exists, or is its rarity likely to maintain over time? Yeah, just how does the trajectory of growth in the cohort compare to previous identified genetic syndromes?
I know who should answer that.
Hi, my name is Christian Schaaf from Heidelberg University. I'm a geneticist and I specialize in rare and ultra-rare neurodevelopmental disorders. And I can tell you, kind of based on our own experience, we discovered 3 conditions between 2013 and 2015. And the trajectory is very similar to one another. So Hao-Fountain Syndrome was first reported in 2013, and we are currently in touch with around 400 families. And then 2 other conditions. One is called BBS-OAS, and one is Hao-Fountain Syndrome. We're in touch with around 200, 250 patients each. The trajectory is similar in a way that we start with a handful of patients, and it takes a while to identify the next handful. Then at some point, once the publication's out there, the number kind of keeps increasing quite rapidly. So kind of between 10 and 100, it goes quickly. But then it tapers off and kind of becomes a steady flow of additional patients who get in touch with us. I think the 250 obviously not a reflection of how many patients are out there. And one of the reasons why it kind of tapers off is because at some point, for USP7, and I'm going to talk about this in half an hour, there are now at least 3 kind of clinical papers out there with increasing number of individuals that have helped kind of redefine the clinical phenotype and understand kind of the spectrum of symptoms. That doesn't mean that all the symptoms have been described and represented kind of reflecting reality, but there are publications out there, and when a new individual is diagnosed and the family comes to get genetic counseling, and they get the diagnosis kind of reported by the clinical geneticist or genetic counselor, there's literature out there that they can base the information onto. And it seems that at this point, with some information out there, some publications out there, a lot of the physicians and families do not feel a critical need anymore to get in touch with us or the foundation, because they have something that they can base kind of their decision making onto. This is completely understandable, but in a way, it's also sad, or it kind of inhibits our future research, because our future research would be much more powerful if we could base it on 5,000 or 10,000 individuals rather than 250. And each one of those junior investigators in the room here, when they write their applications to the NIH to get R1 funding, like they frequently criticize or question for why the NIH should invest $500,000 or $1 million into research that only affects 200 individuals worldwide, right? So it does make a difference there. And so I think the efforts by the foundation and Combined Brain and others are really important, and each and every individual kind of makes a meaningful contribution. And also we realize that what we believe is the clinical spectrum of Hao-Fountain syndrome, for in some areas, may be a good reflection of what it really is. But in other areas, like we are far from kind of understanding what it really is. Like one important point being that, and that's also part of the natural course, the initial cases reported are notoriously more severe than what reality looks like. I mean, it's almost like, if you Google any disorder, you'll always find the most severe representations on the internet. And that's also true for the medical literature, because those severe cases were easier to find, right? They were more evident to understand. They were easier to call by the genetic analysts in the laboratory. So for us, it's really important to also understand the mild cases. And they may be in part kind of a reflection of different epigenetic signatures. They may be a reflection of like different kind of molecular mechanisms, and it may help us kind of understand neurodevelopment kind of way beyond Hao-Fountain syndrome. They may help us understand kind of the neurodevelopmental aspects of autism spectrum disorder at large or others. So each and every one counts.
Can I ask a follow-up to that? Do you think that because a lot of people are diagnosed with autism that they don't do more genetic testing? Do you think that's a reason why some of the rare diseases, like if they're not severe cases, that they just have an autism diagnosis, but they never do their genetic testing?
Yeah, I think there's not an easy answer for that, but I think it's based on a misconception, and it's a question that I'm frequently asked, not just for Hao-Fountain syndrome, but for many others as well. That parents ask, my child has been diagnosed with Hao-Fountain and with autism, but what is it, right? I mean, autism spectrum disorder is a description of symptoms. It's not like a rock solid diagnosis based on like a molecular understanding. So like for your kids who have, like some of whom or many of whom have also been diagnosed with ASD, I would say the diagnosis is Hao-Fountain syndrome, and just like hypotonia and developmental delay are symptoms of Hao-Fountain, the autistic features are also part of Hao-Fountain syndrome.
Yeah, because that's what I think about all the time with Ella, because she was just diagnosed with autism, but it was still part of Hao-Fountain. But would I, because we had so many other things before that, but would I have gotten genetic testing being so expensive and the cost, when there's so many, when autism is so prevalent? And it's just really a symptom. That's what I try to explain to people. I'm like, that's a symptom of Hao-Fountain syndrome. It's not, like just her diagnosis.
Right. And it makes a big difference as to whether you can get genetic testing paid by your insurance company or not, right? And there's differences between like insurance companies in the US. There's like major differences between various countries. Like, as many of you know, I moved back to Germany in 2019, and exome or genome sequencing were not paid for by the insurance companies at that time. We fought hard and like reached a point as of last year that every child and every adult individual too with a rare disorder can get a genome sequencing at no cost, no matter your insurance, whether it's public or private insurance. But like the regulations are very different in different countries. And like with autism, like one thing that really matters for the insurers is like, what is the likelihood that a molecular diagnosis can be made? And oftentimes they kind of, the general concept is like, if a priori there's a 10% likelihood of us finding the underlying cause, then the insurer will pay for it. So oftentimes we have to make that argument that the likelihood is like higher than 10%. And with autism spectrum disorder, if you have autism spectrum disorder with cognitive impairment, so developmental delay or intellectual disability, the likelihood of finding the underlying cause is much higher than 10%. However, if you have high functioning autism, what previously used to be called Asperger syndrome, the likelihood of finding the underlying cause is less than 10%. But like for the insurer, you know, it's hard to kind of differentiate and discriminate. They have like a category that's called autism, and like whether it's marked as like diagnostic rate below or above 10%, like in their documents, we don't know. But like if they just label it as autism and base it on high functioning autism, then yes, it's less than 10% and they may not pay for it. So we have to continue kind of fighting for this and also continue kind of educating the explorers, the insurance companies and other stakeholders also, like in politics, etc.
Yeah. Just one comment on that, actually. It's also how much the disparity in medical care really count for missing several patients. In particular, I'm thinking of rural areas, right? I come from Amarillo, so I wonder whether or not the patients there have the same opportunity to get their kids.
Our experience with that, we would have never found out because they did the first initial testing and they said they didn't find anything. It's just autism. And so like, well, that's just kind of, we're not exactly happy with just that. So we'd like to know more. And the insurance company said it was going to be about $6,000 for the next testing. And then, and so we had to make that decision. We decided, okay, we're going to do that. Then we found out that we could go directly to the lab, which has a sliding scale of cost based on income. And it was still, it ended up being $1,500. And so as far as educating the people on how things work, you know, there's a big difference between 1,500 and 6,000, you know, to most households. So I don't know how we educate people to be able to look beyond just the, hey, this is it. Like, okay, why? And how do you get people to ask why? And how do we make this happen? But we would have never, I'm in the insurance business, so I happen to know there might be, you know, some other ways to get what you need. But it's not everybody, you know, keeps asking why. And so, but I don't know what we can do to keep educating people, but we would have never known if we didn't ask why. And then to find out that, you know, it was significantly less if we avoided the insurance company and just went directly to the source to get the same end result. So that's all.
My name is Kathy Mason and I live in Georgia now. We lived in Charleston, South Carolina area for 8 years. And we pretty much got the diagnosis through the Spark. Now, I don't know if any of you all have heard of it, but basically it's free. You do a swab test and then they collect your sample and then they notify you if there's a match for the USP7. It's the Simon Spark Foundation, but it's through MUSC in Charleston area. And I say that because my son is sitting out there, he's 23 now. When he was in 2015, we did the blood work, which cost $4,000 from Greenwood Genetics. That's in North Charleston. And I think they had a grant from Autism Speak. They kind of paid for it. And it turned up with no result. But then after the Spark, they kind of explained the difference between the tests because as a parent, I didn't really know the difference. I'm like, genetic testing is genetic testing to me, but the chromosomes versus the genes are a different ballgame altogether. So I guess the one thing is though for Spark, you have to have a diagnosis of autism. So I don't know if it would skew your results because everybody that comes in Spark has an autism diagnosis, whereas you wouldn't be getting the referrals from the doctors. I learned on the orientation that basically parents get referred to doctors. They say, oh, do this genetic test. No, James's doctor, not one, suggested genetic testing for him. So it wasn't like brought to me by a physician, an MD physician. I had to go and just search out things. And only because I lived in the area, I knew it was available. I live in Georgia now. Nobody's ever heard of that. You kind of see it on Facebook. It might pop up here and there, but I just kind of wanted to mention that it's free. They'll send you a kit, but you do have to have the diagnosis of autism.
Yeah, I think that's useful. Hi, my name is Jenny Lyday. We are from Indiana. Our daughter Macy just got a diagnosis 2 weeks ago. And so I wanted to kind of talk about that. Our neurologist tried to do prior authorization through insurance, which denied it. And then there's like 3 different tests that you do, and they approved one, but then accidentally ran all 3. So they billed our insurance $53,000 because mom and dad also did some testing. So essentially the doctor's office had to call and say, you ran all 3, you should have only ran the one you got the prior auth for. And then I had to appeal and say, we had prior auth and I had to argue it and I won my appeal. But is there a company that we could pair with, like a genetic testing company? Because I got this packet of like 10 pages, but I don't have any of my raw data. And I would like my daughter's raw data. And I would also like, now I'm a nurse myself. Now I'm curious about her sister, not that she would have it, but something else. I want to get genetic testing on the sister. So I've been researching private pay genetic testing. So could we partner with a company or promote a company where people could private pay, because going through insurance was a nightmare?
I've got something just from our experience. Sure, thank you so much. Yeah, so I mean, we ran against the same thing. I think we had the chromosomal testing. I mean, we're very fortunate again to have Vanderbilt Developmental Children's Medicine, wonderful organization, but due to demand, again, at appointments, they were scheduling people 9, 12 months out for your appointment, and we were able to be there to answer the phone call if they had an opening slot or something, or know who to talk to try to get in. But again, it shows you the difficulty of getting to this point of having to navigate some of these systems. And so they ran the chromosomal testing and didn't show anything. We're recommending possibility of the exome testing. And again, yeah, we knew that was a great expense and insurance denied it originally, but they recommended we reach out to the lab directly. We did Baylor Genetics, was the lab that ran it in Texas. I assume it's somewhat affiliated with Baylor University. I'm not positive. Oh, okay, Houston. Okay. So yeah, they have a financial assistance program. And so we sent off and applied to that. And yeah, basically it was a small copay. I think it was covered completely. I was like, it was definitely like less than $100. So yes, yeah, so they covered it completely. So that is definitely someone, a lab that people should reach out to. Yeah, at least while they still have the funding, whatever funding they have to do this, definitely people in this room that are interested should go and take advantage of that while it's available, because it definitely helped expedite, our diagnosis. And if that wasn't the case, I mean, we wouldn't be here today because we'd still be wrestling with the insurance company and everything else. So yeah, Baylor Genetics, looking at their financial assistance.
Yeah.
I think one of the challenges of teaming up with a lab is that all of you are here because you already have a diagnosis, right? And also even if the USB 7 Foundation teamed up with a genetics lab, it would be post hoc, right? So people need to get diagnosed in order to make the diagnosis to get here, and that's one of the main challenges, because depending on where you live, who refers you to a geneticist, that geneticist would typically send his or her samples to a lab that they've teamed up with, many years ago. Like I used to work at Texas Children's Hospital, Baylor College of Medicine in Houston for 13 years. And obviously, like we would send to our own lab because, it was all kind of one system. So I think that makes it hard, like even though the idea is appealing to team up with a provider, but it's like realistically, it's impossible. If like you have a variant identified and want to kind of follow up whether additional family members carry that variant, that obviously is kind of different ballpark because that's a very targeted analysis that would only be done by Sanger sequencing, which is very inexpensive. And there would be the opportunity to team up with labs and like potentially could even find a research lab that offers that at no cost. Like the challenge in regards of like testing unaffected individuals, especially if they're minors, is kind of their right to know and their right not to know and their ability or inability to make decisions for themselves prior to being an adult. So if like a sister, for example, of an affected individual is clinically unaffected, we would not see the indication to test her for the familial variant, and we would kind of violate her right not to know unless she's an adult and can make decisions for herself. So this is something that we would need to discuss. On the other hand, you also asked about ownership of genetic data. I'm more familiar with the German legal situation now than like the American, but I actually think there's no major difference in that regard. But like you are owner of your own genetic data, and as a parent of children, like because you're basically their guardian, you're also the owner of their genetic data to some degree at least. So if they've undergone exome or genome sequencing, like you should be able to request the raw files. The lab may charge for those, and like we have requests like that in our genetics lab on a regular basis because there's like time and effort and hardware involved. Like we charge 150 euros to like pull all the genomic data, which is like terabytes of data, obviously, onto a hard drive. But we would then kind of give the hard drive out to the family if they request us to do so, because like legally you are the owner of those genetic data. And then like one more aspect I wanted to comment on is kind of disparity. So obviously there have been many studies done on this. White people are more likely to get referred to kind of tertiary providers and geneticists, and minorities are less likely to get the genetics care and genetic testing compared to white people. That's been proven. And then also it does not only depend on that, but also heavily depends on where you live. And it's been mentioned that there's a lot of people with Hao-Fountain in East Texas. Now, why is that? That's in part because of Texas Children's Hospital, Baylor College of Medicine, where there's more geneticists than anywhere else in the United States. Like we looked at the number of board-certified clinical geneticists like 5-6 years ago in Texas, for example. And I think at that time there were 38 board-certified geneticists in the metropolitan area of Houston, and there were only 5 in the rest of Texas. So if you go kind of towards the west of Texas, like there are areas where you can drive 3 hours into any direction and will not meet any clinic geneticists. So it's part of that.
Hi, good morning. I am Olga. I am a parent. I am in Houston. And our testing, after a lot of fighting to get tested, was on through Baylor, Texas Children's Hospital. And when my daughter was diagnosed, I brought up like they wanted to test us as parents, they were not charging. I guess it's more part of their research part into the study and seeing if we were carriers. We were not. But I also brought up she had a fraternal twin that I wanted her to be tested, and they tested her too. I mean, they didn't charge us. Again, does it happen? It's because it's Baylor Genetics Lab, maybe, because they are into research of it. So that's very important to know. And even though we were in Houston and that's where there were papers about this, no one in Texas Children's knew about this condition. And when she got like her brain imaging and they gave me all these bad possible differential diagnoses, and of course not Hao-Fountain or USD7 at that time, and it was horrible and we went through the testing. I was very upset that even in the hospital where there was a paper from a researcher, none of the radiologists knew about these findings of less myelinization in the brain. So even within the systems, not everyone knows what's going on. How that can be helped, I don't know. I'm also in the healthcare field, so I don't know how that can work. But it is us as parents or patients that we need to advocate for our kids and advocate for other people and teach them that they need to ask questions and be pushy and not get any, just like, it's nothing, or it is just autism, or it is just this. We need to teach others. I'm also now found out I also have a rare disease, not related to this. And I spend a lot of time teaching people, you know, they need to find out more. My patients, oh my God, and they want to pull me out of the rooms because I am trying to help them find ways to help them. And I'm like, why their pediatrician has not seen this is obvious in this kid, and why I am the one telling the parent, go ask these questions or call this person. So I think it's also a responsibility after going through all of these difficulty to find diagnosis that we should help others with this.
Hi, my name is John Hoffman. We're parents of a 26-year-old son, and we live in Houston as well. But what struck me was identifying you need the genetics test run. We thought our son had all kinds of doctors growing up, Texas children. Texas has a very large teaching hospital community, but we didn't know you should check the genetics. And it wasn't until he was 24 that somebody referred him to a doctor at Baylor College of Medicine. And so I thought it was interesting, the one in the rural area. And it's just, I think Hao-Fountain Syndrome, organization like this can do a lot just to get the message out to do genetic testing.
And maybe just to comment on that, we need to appreciate, right, the great advancement of science that really allows now that we can do that, right? 20 years ago, this maybe was not even so accessible. It was much, much more expensive. Even data analysis, you can imagine how much data is there, right? So with all of this, gathering the knowledge, improving basic science from biology to computational biology really helps back community, all of us, right? And your children as well. So I think that's really amazing to see, right? And also, I don't know, I don't know how you feel, but it's really important to for patients that you finally figure out what is the bottom, right? And genetics is in a way looking, right? So even though maybe we don't have a treatment yet, it seems like I can say from my family perspective, it's a relief in a way, at least, yeah, there is something, right? And then of course, a lot of things to do, but so I wanted to also trigger some of more maybe research questions if there are any, and then I think we will go back to more clinical again. But I'm not.
And after we're done here, we're gonna have a little break. We have some snacks and all that.
Yes, ma'am. We have a couple of questions here and then coffee. Thank you. I guess mine is sort of in the middle, with question to both Boris, who presented his talk, and so talking about viruses, maybe Christian, sort of knowing more about patients and the history. So Boris presented an interesting talk about how antiviral defense can depend on USP7. And we've actually seen something, I guess, opposite for herpes virus, for USP7. So it actually was sort of pro-viral, the use of USP7 to do it. And just because USP7 is involved in so many known pathways and has so many partners it interacts with, I wonder what is the effect on, other diseases? Do we see that these patients, the USP7 patients, are more predisposed to, say, cancer or, herpes infection or less? Is there anything that we know, or if not, can we get that information somehow from the patient?
Very good question, yeah. Christian, do we start with you?
Thanks, Rita. That's the whole dimension that you raised. And that's something totally known in the molecular biology field, that you can have an impact on the one virus, another virus. And we are in a field that stacks also on regular primary immunodeficiency, where you are forming innate immunity. Yes, like more virus specific, but talk about like intrinsic antiviral effect. And that's a field to build actually, because what I realized, like in the field of biology, when you look at like this specific interaction, that's much less characterized because it's kind of like too specific, and we don't know, like, is it like it has consequences in human population, and that's where I think we have to connect between like the genes that are involved in genetic diseases to better like understand this interaction. Regarding like the role on USP7 and herpes, you're right. So that's the whole literature of USP7 promoting like herpes infection as a cofactor. And I think it's also like some like reflection we have in biology. The viruses that are human viruses that evolve in humans, like herpes, they are very adapted to humans, so they are able to hijack like cellular factors for their replication. And zoonotic viruses that come from animals that are not adapted to humans. For example, West Nile, there is no human adaptation because there is no human-to-human transmission. That's where you can identify these antiviral factors that actually are antiviral because the virus is not adapted at all to interact with this sort of like barrier. And there are examples that are starting to be raised by genetic factors influencing viral infection. There are some cases of like NCP1, I'm not sure about like the disease, like is it a protection for Ebola infection, for example. So that's a field to build to understand all of this interaction to have more connection. And beside that, another layer is the layer of the interaction between diseases and immunity. And that's also new, just starting to connect, for example, genetic diseases involving metabolism to actually like immunity. And start to like screen like which genes involved in metabolism has an impact on immunity. And that's what we want to do with what genes are involved in virus-host interaction, involved in genetic disease, have an impact on infection.
So, and also, I used to work in a lab that studied spinal muscular atrophy, which is another genetic neuromuscular developmental disease, call it. And so, because you guys have a number now, this is going to help so much, because what we did with spinal muscular atrophy is that is something that the parents have to be carriers to get for the child to get it. And so what we did is because SMA has a code, you can get huge data sets from insurance companies. And so this was done when I was in Boston. And so got all of this data. And then what we could do is use the code. So we found the children that have a code for SMA, and then you could link that to their parents. And so what we found is that just being a carrier, so the parents had, were more likely to have cardiovascular defects. a neuron muscle disease. So I think that's going to really help with these questions about infection. And it's just all in the insurance coding and getting more patients involved. And then you have these huge data sets you can find. There's a company in Boston that is completely focused on this, that was spun out of this research actually. And so I think that the patient group might be too small to pull out some real significant differences. I mean, as a clinician, I'm sure you maybe agree or could comment, but I think that's really going to help. I think that's gonna be huge.
I fully agree. I think the ICD code will help. It'll take some time, for that to be implemented and documented, but then eventually, kind of large hospital systems like Geisinger or Kaiser Permanente and others, like you could then kind of pull information not only about the prevalence of Hao-Fountain, but also about kind of comorbidities occurring and whether they are statistically significantly different from like non-Hao-Fountain kind of patient cohorts. Fully agree. And regards of kind of virus susceptibility in Hao-Fountain, I'm not aware, but this is a question that we've never asked in our kind of questionnaires and phenotyping studies. So certainly something to kind of follow up upon. And then regards cancer, I mean, there's obviously abundant literature about USP7's role in cancer, mostly in regards of like amplification or overexpression of USP7, which like oftentimes that question, like when families are newly diagnosed and they kind of go online and go on PubMed and search for USP7, this is what comes up. And it's like an early question that we ask, like, does my kid have like a higher chance of developing cancer? Like most of the kids with Hao-Fountain syndrome, have loss of function of USP7. So if there was kind of a risk adaptation, it would mean that kids with Hao-Fountain have a lower risk of developing cancer rather than a higher risk because they have less USP7 function and not like an overexpression or higher function, what's mostly reported in the literature in relationship to cancer. But again, this is fully speculative at this point.
We can have as well what we expect to have as an outcome to emerging this field of infectious disease and rare genetic disease is able to reach other places and other areas. So then when I show you viruses of disease happened in Latin America, the field of virologists working with these virologists are connected to hospitals in Brazil, in Colombia, in South America, in the islands. So that's also something we'd like to promote. Like, if that's a risk factor for infectious disease, for example, we want to know it during the outbreak and during the epidemics, because we know that only a minority of people that get severely impacted by this infection. And if we have any way to reach out, like this hospital in Brazil said that actually we should care more as comorbidities, having a lot of pre-existing condition, development, and we know that this gene is important, that could help also maybe take diagnosis for them and have this connection.
Okay, is there any other question now? Otherwise, maybe we can have a little. Okay, of course.
Hi, everybody. I am Diana, my husband Laurent. We have two child. Nina is Hao-Fountain since this year in Canada to Saint Justine. After 15 years. Yeah. Sorry for my bad English. I'm Belgium and my first language is French, so I can try to do my best. Thank you. I have a special question about some disorder. Nina is the second time that she has an extra teeth on the palate, the mouth, and it's very hard because we have to
do a surgery.
Surgery, and before without the back.
I don't know.
Before the surgery, this iteration.
You need to wait a lot of time because they don't have space directly.
Yeah. And she has a lot of pain, and she pulled her hair very hard.
Alopecia.
Yeah, very. She doesn't have hair.
It's a little bit like me.
Yeah. She looks like. And we would like to know if it's an incidence, a link with the Hao-Fountain, because there is a lot of extra teeth in her mouth. Thank you. Hi, so I introduce myself as a mom, but I am a dentist. And all those questions get like sent to me. And I'm talking about teeth tomorrow, and I don't wanna repeat or spoil what we're talking about, but basically we don't know. Things that are not studied are not known. I keep tabs of the dental stuff because I'm curious, and things have happened through even taking care of my own daughter. I'm trying to get you taking care of my daughter that has sparked questions with us. And then you have to understand like these things as more teeth, less teeth, enamel defects, and all this stuff, they are common, or I should say they are rare or less incidence in the general population. So we cannot say without having any research, right, that this is a Hao-Fountain thing. So each time there's a question that says, oh, if this is a Hao-Fountain thing, and I'm like, we cannot blame it. We cannot blame things our kids have on the condition because they are just maybe happening, okay? So if you have any pictures or x-rays or anything that you can share with me, I can look and give you an idea. But yeah, so I'll talk more about it tomorrow. I know recently someone asked about anesthesia and because of the disease, they could not get these. I think it was really like probably like a loss in explanation of what the dentist was thinking. I think it's more because the dentist was concerned about the airway of the patient and how he felt safe of doing it. And I'll talk about that again tomorrow.
So basically, because Olga is doing kind of a study on teeth in relationship to Hao-Fountain syndrome, all the questions regarding teeth kind of forwarded to Olga. She's really an expert. Yeah, and we do it. Yeah.
Tomorrow.
Sorry. I think I know.
Anyone else? Otherwise, I would like to thank you. I think it was a wonderful discussion and on both clinical and patient as well as the research side. So thank you for participating and enjoy the rest of the meeting.
So we can take break. Come to like 15-minute break. Is that good for everybody?
Did everybody hear me in 15 minutes? Oh, it's like 11:05. Be back.
I did, and that's why I brought it, but I love it.